Setting up the next speaker, I will introduce her. Dr. Lena Jones. Dr. Lena Jones is a professor and Buchanan Wilson Chair in Eukaryotic Cellular Signaling at the University of Guelph. She completed the PhD studies at the University of Toronto and obtained postdoctoral training at the Lunenfeld Tannenbaum Research Institute with Dr. Tony Paulson before recruitment to Guelph in 2006. Her research investigates fundamental mechanisms of cell communication by phosphatidylate adapter proteins, and she has made significant contribution in understanding how such signaling pathways become deregulated in prevalent human pathologies such as kidney disease, cardiovascular disease, and cancer. Dr. Jones has received numerous awards through her career, including a CSAHS Crescent New Investigator Award, an Ontario Early Researcher Award, Answer University Faculty Award, and the John Charles Polanyi Prize, and she was recently named as a member of the Royal Society of Canada College of Scholars, Artists and Scientists. Dr. Jones also received the inaugural Graduate Student Mentoring Award in her college, as well as a Kidney Foundation of Canada Merit Award for Outstanding Community Partner, and she was named YMCA YWCA Woman of Distinction. The research of Dr. Jones is supported by CSAHS, NSERC, the Kidney Foundation of Canada, and the Cancer Research Society. I wanted to tell you.
Okay, well, thanks, Francesco. I know those are some words you don't use in your everyday research, so I appreciate you giving that nice introduction. And it's a pleasure to be here. I've been part of the CCVI, I think, since it started. And what my group brings to this is something a little bit different. We work on the kidney, and the kidney is part of the cardiovascular system. So I hope to be able to show you that today. And I thought it was an interesting idea to put Todd and I together in this session, because we actually both have labs a couple of doors away from each other, and we have never worked together. And I think after you see what we're doing and what Todd is doing, that we should be working together. So I'll thank Temi for making that suggestion. So what I'm going to talk about today are signaling pathways regulating development and function of kidney vasculature. So the kidney is something that we've worked on for several years now. But my interest in cardiovascular disease and cardiovascular development really started as early as my PhD. So during my PhD, I worked on blood vessel development in the growing embryo. So that's shown up there at the top. So we looked at sort of how that embryo is patterned. Then I went on to look at a little bit more about how these blood vessels would be important in feeding a tumor. So in the process of tumorigenesis and angiogenesis, I continued my studies there. And then more recently, and for the duration of the time that I've been at Guelph, we've been looking at the kidney. Sure, I should have looped this movie, but I didn't. Okay, so here's a vascular cast that I actually did as a graduate student before I knew that I'd really be working on kidneys for a living. And what you can see is that the kidney is a highly vascularized organ. So the kidney receives about 25% of the cardiac output, and it serves to filter our blood. So one of the main functions of the kidney, it has several others, but in the context of this talk, the main purpose is filtering of our blood. And that happens out here in these structures known as glomeruli. So these glomeruli are part of the nephron. And each of our kidneys, we have two of these, each of our kidneys has about 1 million glomeruli. So these are really important structures that function in that filtration process. So just to break that down a little bit more, here's that paired organ structure. So they're located on our lower back. And I mentioned that we have these filtration structures located on the outer cortex of the kidney, known as the glomeruli. So what these guys do is they bring the afferent arteriole will bring in the blood. It'll pass through this capillary knot, which is basically what the glomerulus is. And any of the filtrate that passes across, so waste and some water that comes across, will be collected here in Bowman's capsule on the outside. And that's the primary urine, which will then move on through the series of tubules that we're familiar with, which will concentrate the urine. So what you can see is that these glomeruli then are filtering structures. So that means they need their filtering structures and their capillaries. So this capillary bed has several pores in it, and that's what's shown in this next diagram down here. So this is a sort of a subset then of what a portion of that capillary loop would look like. So as with any vascular bed, the lining is endothelium, but in this case it's a fenestrated or porous endothelium. That's important in filtration. On top of that, in purple there, you see the basement membrane. And on the outer surface of these capillaries, you see these really unusual shaped cells known as podocytes. So the podocyte has these extensions that come off it, which terminate in something known as foot processes. So these foot processes are shown here. They're like little teeth-like structures that come off the primary process. These are highly actin-rich structures, and they wrap themselves around the entire capillary. And it's these podocytes then that form the final layer of filtration in this glomerulus. And if we look now in, again, higher detail, now looking between a few of these actin-rich projections or foot processes that I just mentioned, what you'll see is between them, there's a molecular filter, which is known as the slit diaphragm. So it means then our blood is passing through first the endothelium and then this molecular filter, the slit diaphragm, which is sort of the terminal stage in that filtration process. So just to orient you here again, we have this endothelium, the overlying basement membrane, and the foot processes with that slit diaphragm between them. That slit diaphragm is a complex of molecules, and I'm going to focus on that for most of the talk today. So what I hope you can appreciate from this diagram is that the podocyte is a very unique cell. It's unique in terms of where it's located in the body. We don't have podocytes anywhere else in our body. It's unique in how it has to function. It has to function in filtration, and part of that function is determined by its shape. So it sits up here like a sort of a plump-looking octopus with these projections coming off it. So all of these things are unique about the podocyte. Further to that, what you can sort of appreciate from this cartoon here is that the podocyte would see a lot of different stresses in the body. It would see shear stress, for instance, from all the flow passing across it here in Bowman's capsule. It also sees shear stress here, where you have the filtrate pushing through these structures, and also the tension that the basement membrane will be subjecting with every beat of the heart. So these podocytes need to be able to respond to these different challenges in the body, and much of that is dictated by this actin cytoskeleton that they have, and their ability to sort of dance around these changes that are happening to it, as I said, with every beat of the heart. So the plasticity of this actin cytoskeleton is what really makes the podocytes able to sit in this part of the body and withstand all of these challenges. So just to sort of orient you now to some of the images that you'll be seeing here, we have the podocyte up here. This is a scanning electron micrograph of that. This is a transmission electron micrograph, so now you can see all those teeth-like foot processes, and this electron-dense line between them is the slit diaphragm. In cartoon form here, we see these processes. They need to be anchored to this basement membrane in order to withstand all of these stresses that the blood and shear flow are applying to them, and this is the endothelium. So what happens now with podocyte injury or in kidney disease is that we have a flattening of these cells. And that flattening is sort of depicted up here. We also see what's called simplification of these processes. They retract and they're not able to properly filter anymore. Part of the adaptive change is for these foot processes to flatten out across the podocyte, across the basement membrane. We still don't understand a lot of what's happening here. But if we look in sort of a cartoon form of what might be happening, these foot processes are flattening out. This is sort of an attempt to hold on. But at some point, this is a point of no return for these cells. And what happens is you get detachment. And when we have this sort of effacement-detachment transition, we start to lose the proteins that are supposed to be in our blood. They start to pass into the urine. And that's a situation known as proteinuria. I'm going to show you some of that today. So what's a remarkable fact about kidney disease is that it affects about 10% of the world's population. So chronic kidney disease is sort of, again, this point of no return. And 10% of our population is hundreds of millions of people around the world. This is 20 times more than the number of people who have cancer. So kidney disease is really one of these silent killers that's very much underappreciated. Furthermore, the treatments for kidney disease are virtually, we don't have any, basically. So we don't have any treatments for kidney disease other than things like dialysis or transplant. And these sorts of therapies, if you will, are unsustainable. So in the absence of any real targeted therapies for kidney disease, there really is intense clinical interest in trying to sort out what pathways might be important, signaling pathways in leading to this disease, so that we can look for targets to design better and more effective therapies. So that's really where the research in our lab is coming in, looking at the cellular signals that are important in setting up these podocytes and what goes wrong when these podocytes start to detach like this. That's what we're interested in. So one of the proteins that we spend a lot of time studying in my lab is this transmembrane protein known as nephron. And nephrin was first identified about 15 years ago now as the cause of something known as congenital nephrotic syndrome, when babies are born with kidney disease. This is where they first found this really important protein. But we now know that it seems to contribute to maintenance of this filter. So when we have changes in expression and function of nephrin, we also see kidney disease. So nephrin is located up here in the slit diaphragm. It's actually one of the major portions of this molecular filter. And just to show you some key features of what's happening here with this protein. So here we have the extracellular region. It spans that filtration slit here. So this is how it will be subject to that shear filtrate flow passing through it. But the way in which it is able to maintain the shape of these foot processes is through its anchoring to the actin cytoskeleton. So this protein here, nephrin, is connected to actin. Furthermore, what's important in allowing these podocytes to remain adhered to that basement membrane are these structures down here known as focal adhesions. So these contain proteins like integrins, and again, they have a dynamic response in how they maintain adhesion. So several years ago now, we became interested in nephrin for the reason of these tyrosines that it has on here. And as Todd already mentioned, proteins can become phosphorylated. Tyrosine is a key target for phosphorylation. And one of the things that we noticed was that 3 of these tyrosines have a very similar sequence. And when they become phosphorylated, it sets up binding sites for a class of adapter proteins known as the NCK adapters. And at the time when I was doing my postdoctoral fellowship, this was a protein that I was studying in the context of a different cell system. So that brought me into the podocyte. So here we can see that these are molecular adapters. They have no enzymatic function, but they have a domain that allows them to specifically recognize changes in tyrosine phosphorylation. They also have a series of SH3 domains which allow them to bind proline-rich sequences and target proteins. So we're bringing up all this cargo up to the membrane of the podocyte by virtue of this SH2 domain binding to the tyrosines. And there's 2 of these NCK proteins, NCK1 and NCK2, and if time permits, I'll tell you a little bit more about that. But what these proteins do is basically connect proteins like nephrin to the actin cytoskeleton. So several years ago now, we devised a system where we could activate nephrin in the membrane akin to it sort of being mechanically clustered, and looked at what happens to actin in these cells. And we did this with a nephrin molecule that had all of its tyrosine sites or had them mutated to phenylalanine, so it could no longer recruit this. And what we found using cells that don't have any NCK, so these are null fibroblasts, is that when we have nephrin shown here in green with NCK2, when we cluster these proteins, they come together in sort of this turquoise colored spot. And associated with that, we see these comet tails of actin, indicating that where we have a nephrin and an NCK cluster coming together, we have sort of localized generation of actin filaments. So indicating that this signal could be important in regulating that actin cytoskeleton. And we further went on to show that you need these tyrosines for this effect, to make these actin comets. So one of the strategies that we use in my lab to understand physiological processes is to generate gene-targeted knockout mice. And so in the case of NCK, this is challenging because there's 2 of these proteins. Individually, they're viable, but when you knock both of these proteins out in the animal, they die during mid-gestation due to gastrulation defects. So these are not able to properly form an embryo. So to get around this, we use the Cre-LOX-P system, where we're able to insert LOX-P sites around one of the exons in NCK2 in this case, and using a tissue-specific Cre, we're able to then make a conditional knockout in a tissue of interest. We've done this in many cell types, but I'll tell you today only about the podocyte. So when we did this, we generated podocyte-specific NCK knockout mice. These mice are born, but we noticed as we were starting to wean these animals that they're failing to thrive. They're much, much smaller than their littermate controls. And looking at the podocytes in these animals now, if these are the control cells, you see these nice plump cell bodies with these projections sticking out from them. When we did the NCK deficient ones, these cells completely collapse around the basement membrane. And the transmission electron micrographs told us that they don't even make foot processes. So this is a really, really profound phenotype. We looked at the urine in these animals, and as I mentioned, proteinuria is something when you have leakage of the barrier. We can see that here in this blue Kumasi stain. But more recently, we've gone on to also show that in that urine, we're seeing podocytes actually falling off. So we're able to see podocytes in the urine of these animals, indicating that in the absence of NCK, we have a detachment phenotype. So a very pronounced phenotype here. And we also went on to show, using another mouse model, an inducible system now, when we now delete NCK from adult podocytes, so we have nice plump podocytes, and then we delete NCK, they flatten out as well. So it indicates that these proteins are really important, not just to generate the cell, but also to maintain it. So to understand a little bit more about what these cells are doing in the body, we've had to now go to a cell culture system, as Todd described as well, and we've generated immortalized podocytes that we can grow in the lab. And I won't show you anything more than this to show you that this is actin staining in a wild-type cell. In a wild-type podocyte, you have this nice cortical actin structure, but in the double knockout cells lacking these NCK proteins, the actin is completely disorganized into these stress fibers. And looking at the adhesions now in these cells, those focal adhesions, those integrin complexes that I told you hold the cell onto the basement membrane, you see it in the wild-type cell, they're only on the outside. This is where these focal adhesions are normally decorated adhered cell, but in the case of the NCK knockout cells, they're all over the place. Again, highly disorganized actin in these animals. I'm going to pass through this slide here because, wow, time is going fast. But this is simply to show you that we have two NCK proteins, and they seem to be individually, they're not, we can live without them, but when we challenge an animal, you need both of the NCKs. So now we turned our attention to knowing that NCK is critically important in the podocyte. What about that upstream signal? What about nephrin? So this is sort of the culmination of many years of work in my lab. But we asked the question, what happens now if we take out these tyrosines so that we can no longer recruit the NCK protein? What happens in animals lacking nephron phosphorylation? So we call these animals our nephrin Y3F for mutating the 3 tyrosines to phenylalanine. And I wouldn't be showing you this if there wasn't an important phenotype. So what we see in these animals is that a couple of weeks after birth, they start to spill protein, and these animals here progress throughout their lives. They're not quite as severe as the NCKs, but they do develop kidney disease, indicated here by this proteinuria. And if we look at the foot processes in these animals now using electron microscopy, you can see this is the wild type here. This is an older animal than what I showed you earlier, but here we have these sort of funny shaped projections in these animals. So the foot processes are not forming properly, again in the absence of nephrin phosphorylation. And I should also sort of pause here and mention as well that we know that this phosphorylation is important because we've been able to look in both the embryo and in the adult and show that nephrin is phosphorylated throughout life. And in human diseases associated with podocyte injury, we see a lack of this phosphorylation. So the phosphorylation is important. It's present all throughout life, and it's gone in disease. So it really suggests that there's a link here between phosphorylation and podocyte morphology. So far we know that we have this important signaling nexus between nephrin and NCK. It's important to maintain the morphology of the podocyte, but what we don't really understand very much about is what are the cellular and molecular mechanisms that are being driven by this interaction. And that's what we're really trying to figure out now. This is sort of unpublished stuff from the lab that we're currently working on. So I bring you back to this diagram here to show you that we know that we have this important slit diaphragm molecule here, nephrin. It becomes tyrosine phosphorylated, which allows it to bind to NCK, which would be this green protein here. But what we don't know is how does this signal help us to maintain adhesion? So I'm going to draw your attention to this side here. What is the link then between nephrin and the focal adhesions? And we believe it's NCK for the reasons that I'll show you in these next few slides. So I like to show this image. This is we have had a recently graduated Western Blot queen leave our lab, and so this is an awesome pile-up of information. But what I want to just show you is when we see the phospho signal for several of these proteins. This is the phospho for a protein in the focal adhesions. Here's another one in the adhesions. This is NCK itself, and this is nephrin up here. So just to orient you to what we're looking at here, on the left-hand side, these would be wild-type podocytes that we've grown in the lab. We've introduced our nephrin clustering system, which can activate nephrin. And when we do that, you see a rapid sort of phosphorylation of nephrin itself, and you see this nice induction of phosphorylation of proteins that would be sitting in the focal adhesion. And when we now delete NCK from this system, we see a flat response. So we actually don't even see nephrin phosphorylation, which is a different mechanism I won't explain. But you can see here this focal adhesion protein and this other one here, CAS. There's very little phosphorylation on these proteins, indicating that you might need this signal from NCK to drive the response from nephrin through to the focal adhesion. So we need phosphorylation here to drive the signal down to the focal adhesion. So we believe this is somehow important in transmitting a force down to the base of the cell. And to test that, we've set up a recent collaboration with a group in Scotland who developed this very unique microscopy technique known as elastic resonator interference stress microscopy. What this basically is a sort of an elastic substance in between two glass plates, and then you can grow cells on top of the glass plate. They're very, very, very thin gold layers in between as well. And when the cell is moving, you can see deformation of that elastic substance and actually record that through several different channels, which are not shown here. So what we've been able to do using this technique now is show that these cells that are expressing wild-type nephrin, before we have our activation of the system, they're sort of sitting here. We're looking at the center of the cell sort of pushing down, and where we see the outside, it's pulling up. That's indicated by this displacement bar here. And when we activate nephrin through this clustering system that I mentioned, you can see the cells pushing down on the middle of the substrate. So that's being recorded by the elastomer here. But in the case of cells that don't have those nephrin tyrosine phosphorylation sites now, you can see that the cells basically sit there. So the cells are not able to transmit that force. And I'll just show you a quick video of this as well. It's pretty impressive. So these are cells that are sitting there for 2 hours. And then when you see CD16 come on here, you'll notice this cell start to push down as soon as it sees the signal. So it's just had this activating system, and it pushes down onto the surface. So indicating then that when you have nephrin sitting in the cell, it's activated to induce its phosphorylation. As soon as we have that signal, the cell pushes down. So the cell is producing force. So we believe then that what this shows is that we have, this is my last slide, what this shows then is that, for a couple of different takeaways from this, that first of all, the NCK proteins themselves seem to be important in regulating the adhesion of these podocytes to the basement membrane. So in the absence of NCK, we quickly transition to effacement and detachment. So we have this very heavy proteinuria in this situation. We've also shown that both of the NCKs are important for this in the setting of challenge. And this is important in our sort of classification of diseases, because we've actually been able to determine that people who develop diabetic nephropathy have changes in the levels of NCK1 in particular. So there seems to be some clinical connection then between these proteins at this point. The other sort of takeaway from this is that we're beginning to figure out a signaling axis that's important in allowing these podocytes to remain adhered to the membrane. So we've hypothesized that it's NCK that's sitting here. It's sitting here to sort of join these two different compartments of the podocyte. So it's joining the slit diaphragm here to this focal adhesion complex, again, two different molecular centers in the podocyte as well. And this sort of touches on what Todd was just mentioning, that we believe this protein nephrin is now playing a mechanosensing role. And in this way, it's able to adapt to the shear stress that's coming through the filter. And in that way, by connecting through NCK now, maintain its adhesion. So we're interested now at looking at whether or not we can sort of experimentally prove the fact that this could be a mechanosensor. So that's where I want to end there. And I'll just stop by thanking the people in my lab that have done this work. There's a couple missing here, but a lot of this work was done by Claire Martin, who's recently left my lab, and Laura New, who's instrumental in generating the Nephron Y3F setting. We've got some great collaborators on campus and off, particularly Jeremy. He helped us with a lot of the injury models, and collaborators on work I won't talk about today. And then the funding sources are listed there. We're also on Twitter, but I'm not a very good tweeter, so anyway. So that's it.